Question 7, help me video. Tough question this. So we're talking about kidney cancer. We've referenced the structures involved in the production of urine. Explain how a kidney tumour could result in blood in the urine. So, with reference to the structures involved in the production of urine, where is urine produced in the kidney? Well, it's in the nephron. Which part of the nephron? Or you can say it's in the glomerulus Bowman's capsule, but which part of that? Exactly where is the filter? And you could then suggest if that's damaged, what would happen if the filter is damaged. Then we have the classic measles and star. And you should know these. This is all A01. So you've got 5 marks of A01, just remembering it, but people have struggled. I'm just going to put a line on there. And everything's heavy, so both strands are heavy. Then we start using the 14N. If we get semi-conservative replication, one strand will be heavy, one strand will be light. But as they're fixed together, then we don't see them at the heavy, and we don't see them at the light. We see them halfway. In the next division, well, this is already half and half, so that splits, and the light half gets another light half, and the heavy half gets a light half as well, so it stays there. And now we've got some light and some medium. The light will just get more light, and the medium will, half of it will be light and half of it will stay the hybrid medium. So you just need to look at your textbooks and notes and you'll see this. Name the theory. Even the people who can't draw the tubes have generally got the theory right. Explain how the results before and after the first division. You're just explaining this first division. You're just saying what we're saying here to make, you're starting off all heavy before and after. So we're starting off all heavy and saying we're building new strands only with light, and explain why this is a hybrid and where the strands have come from. So one strand's our template strand, one strand is a new strand made from 14N. So it's just the theory there that you know about. Part C. Now we've got a protein that is involved in the control of the cell cycle. And this protein, so high levels of the protein block the S phase. Where's the S phase? Here it is. So this protein essentially slows down the cell cycle. So slows down the cell cycle. The question asks about normal cells. So I'm not going to talk about cancer in this part of the question at all. It talks about advantage for normal cells. Why would we want a normal cell having a slow cell cycle? We can just come up with any explanation that you like, really. This is why a cell doing its normal job wouldn't want to replicate but carry on doing its normal job. So that's quite tough. This second part, why do we want cells to have a low level? I mean, this cell cycle will go around faster. Well, there are times that we need fast replication. There's a few suggestions. So when do you want cells to replicate really fast? Not talking about cancer, and that's the answer there. Part 2, suggest why abnormally low levels. So this is now abnormal, and this is our tumour. Could result in development of a tumour. So it's just explaining what this does, this P27. So P27 stops this phase. So your first answer is going to be, well, this goes around faster, blocks the initiation. So it doesn't block the initiation, just goes round quicker, and then you can describe, the tumour, uncontrolled cell division. Part D. This is the protein, and as you can see, if you looked at that, you'd go, it's a tertiary protein. And it even mentions one of the types of interactions. Some confusion here, and this whole thing is an amino acid residue. This whole thing is an amino acid residue, and it's number 88 on the chain. So this will be in here somewhere. Number 88, this is the amino acid, and that's the R group. And as you can see, this is the R group here. But look, it's changed. So we've got a different R group. And so the first thing we need to do is go, wow, look at that different R group. This is how it could change bonding. Look, there's minuses on there. And then you can say, well, this is how I think the shape would change, and mention the level of protein structure. And then go back to, well, this says it acts as a competitive inhibitor. Well, the last mark is, well, how would it not now work as a competitive inhibitor? So just thinking of that theory of competitive inhibition. Good luck.